now we switch to English. So first of all, I would like to thank Bobana and Federica for the invitation and to present this seminar. So, I will first introduce myself. So I'm Riccardo Spizzo and I'm working at Centro di Riferimento Oncologico in Aviano. And I'm running a non-coding RNA lab, and we are investigating non-coding RNA regions involved in cancer, specifically in ovarian cancer and colorectal cancer. And about 2 years ago, since we are working with non-coding RNA regions of the human genome, about 2 years ago, we started using the CRISPR-Cas9 system, since it's very useful for our research projects. And I don't know how many of you know about CRISPR-Cas9. Can you raise your hands? Do you know? Have you ever read something or? Okay. And so this is the outline of my presentation. So I divided it in 3 parts: the introduction, our real life experience. I say real life because also involves unwelcome experiences, not only welcome, because usually when you have a new technology, they show only the welcome experiences, not the unwelcome experiences. And then some troubleshooting. This is based on our experience. Okay, that we are a small lab, we just started, and at the end I will give you also, if you are interested, some references to papers or websites that are useful to start working with this technology. So in the introduction part, I will give first an historic perspective, and here, can you see it or not? So I use, I take advantage of, the screen is small compared to my computer. So I take advantage of a recent review from Eric Lander, published on Cell 2016, where, and it's, the title is The Heroes of CRISPR. And he depicts the history of CRISPR and Cas9 system with this map and also a timeline. So you see that the discovery of CRISPR starts in 1993, actually 1989, but the application of CRISPR-Cas9 system for the genome editing started actually 2012. So it's about 4 years that everything started. And so the beginning was in 1989 when Francisco Moica started his PhD student, and the project for his PhD was to investigate this the Allophorax mediterraneae, and he started doing some sequencing from the genome. And at the beginning, he got these multiple copies of near-perfect, roughly palindromic, repeat sequences of 30 base pairs, and they were separated by 36 spacers. And he was the first to find these novel repetitive elements. After a while, he went to for a postdoc in England, in Oxford, and when he came back, the University of Alicante offer him a position, but they didn't give him funds and space. So they give him only an office. So he decided to do the only thing that he could do with the space and the computer, and so he turned to the bioinformatic part of his research. And at that time, what he was doing was blasting. You certainly know now the BLAST program. So he was blasting the sequencing that he was getting from these repetitive elements that he called CRISPR, with all the sequences available. Since one day, the since these, at that time, the database were not very updated, they were keeping adding new sequences. One day, it came back in a sequence, a perfect match with the P1 phage. But he knew that the sequence that he entered in BLAST was from an E. coli that was resistant to P1. So he had the idea that those repetitive elements were actually a sort of memory of the past infections. So a sort of immunity of bacteria against the phage infection. And so this was 1993, and for about 10 years or so, other investigators tried to prove, and they did indeed confirm, that his idea was right. So the first evidence was that by putting together the phage and the bacteria and generating bacteria that were resistant to the phage infection and analyzing these CRISPR loci before and after the infection, they actually found that some bacteria that became resistant to the phage infection acquired in those loci the sequence of the phage. So what in that moment they said that they actually see the immunity of bacteria in action. Probably immunity is not correct for a bacteria, but it gives the idea of the mechanism involved. The next step was to exogenously insert a sequence of a phage in the bacteria, exactly in the CRISPR loci, and see if the insertion of the sequence will generate a bacteria that was resistant to the phage infection. And indeed it was the case. So that was like a flu shot for bacteria, because it was like vaccinating the bacteria against the phage. At that moment, they knew that these CRISPR loci were transcribed. There was a non-coding part, but also a coding part, so proteins that were called Cas, because were CRISPR-associated proteins that were involved in the process. But so it was known that both the non-coding part, the RNA part, and the protein part was necessary for the mechanism to take action. But they didn't know whether the target of this system was RNA or DNA. And so Marafini in Chicago, this smart experiment, you can, with the CRISPR and Cas, you can target whatever sequence you want, even a plasmid. So they inserted a plasmid, but the target sequence was splitted by an intron. So if the target was the RNA, the CRISPR and Cas system should work, because when the DNA is transcribed, you get the RNA, and then the intron is spliced out, then the target is available for the targeting. But if the target is the DNA, the intron will break the target and the system will not work. And that was the case. So the DNA was the target of the CRISPR-Cas system. And so they defined it as essentially a programmable restriction enzyme. You can decide to target a sequence, and the system will cut exactly between the Cas9 will cut between the third and the fourth nucleotide in the target sequence. So the system was this is the phage, this is the bacteria, you have the double-strand viral DNA. This at some point is processed by the Cas protein, and part of the sequence are inserted in the CRISPR loci, which are transcribed, and also with other Cas proteins. They are assembled with the proteins, and this is the guide RNA, so the RNA part that eventually recognize the DNA of the viral DNA, and then starts to break this apart. And so the infection is prevented. So what about genome editing? so it started about in the 80s with Capecchi, and it was a very powerful tool because you can modify the genome of an organism like mammalian genomes, and to investigate a specific gene or a specific disease. So even though it was very powerful, it was evident that was also very inefficient. It was very difficult to generate homologous recombination in that exactly locus that you want to target. But it was also clear that if you were able to create a double-strand break in that position, that will amplify the chance that you have a homologous recombination for your target. And so the first general strategy were the zinc finger nucleases, but was quite difficult. You had to spend a lot of time to generate the nucleases to target exactly that target. Then they came the Thale that were easier compared to the zinc finger, but still they require some time. So in 2012, it was clear that the CRISPR could be the right answer to this problem, because the CRISPR, as you will see later, is very simple. Okay, it's a matter of 20 nucleotides of RNA in clone in a plasmid. But in 2012, there were the researchers involved in the CRISPR field. They were quite skeptical that the system from the prokaryotes could be translated to be used for the eukaryotes. But then Feng Zhang from the MIT in 2011, as soon as he knew about the CRISPR, he started working on this, because he was an expert in the zinc finger and the Thale system approaches, and he knew how much you have to work to get what you need to do the double-strand break in that region. And instead, he understood immediately that the CRISPR were the right solution. So in less than a year, he was able to adapt the systems that were described, and he published the first paper where he was able to prove that using the Cas9, that's the protein, and the guide RNA, that is the RNA that is transcribed by U6 promoter, he was able in HEC293 cells to target and generate a double-strand break in that target. And after that, I don't know if you ever heard about Adgene. It's a non-profit company that where the researcher can send their own plasmids, and for $65 you can get a copy of the plasmid. So Feng Zhang was so smart to put all his plasmids in Adgene, and in 3 years, 25,000 people asked for his plasmids. And after that, he also resolved the 3D structure of Cas9, and he was able to generate, he just published on Science, at the end of 2015, a new Cas9 that has a reducibility to bind DNA. And by this mean has a lower ability to recognize off-target sites. So he created a Cas9 that is more specific for the target. And also, so Cas9 is only one of the Cas proteins. There are many more, and only now they start to investigate all the proteins. And he also described this one, CPF1, that has some advantages compared to Cas9. For instance, you require a smaller fragment of RNA, not 120 like Cas9. And he also, Cas9 makes a blunt double-strand break. Instead, the CPF1 is a sticky hands, like the restriction enzymes, okay? So the typical application of Cas9 is to knock out a gene. So what you do is, this is your target, for instance. You design this sequence, the blue sequence, that is the target the sequence of RNA that is complementary to the DNA. And by expressing the Cas9 protein and the guide RNA, you create a double-strand break. When you have the double-strand break in that position, you can have two choices. I mean, the cell try to repair the damage, and this eventually can be done by non-homologous enjoining or homologous recombination. If you have this is the more frequent, and is also error-prone. So what it means is that in some cases, the system is able to repair the double-strand break and generate the exact same sequence of the parental DNA, the wild type. But in some cases, you can either, you have indel mutation, either deletions, insertions. You will see later on what you get. And in most of the cases, if this is a coding sequence, in most of the cases, then the indel mutation results in a stop codon nearby or downstream. And so you have the knockout of the protein. On the other case, in homologous recombination is less frequent. And but what you can do with homologous recombination is that if you have a template that contains specifically a mutation that you want to recapitulate in your cell line, you add the template, and by doing the double-strand break and using the template, you may eventually generate the mutation, the exact mutation that you want. So this is the most common application. This is less common because also this is less frequent compared to this one. So this is from the Agin website, and these are the possible applications of CRISPR-Cas9 at this moment. The typical one is the cut, this one, okay? So you generate a double-strand break and with these two outcomes. The other is a nick. So instead of doing a double-strand break, you can do a nick. The nick does not elicit the non-homologous enjoining pathway, so you don't have errors, but you can still have the homologous recombination. So you get rid of all the errors generated by non-homologous recombination, and you can use that to amplify the, not really amplify, but you have an increase in the probability to have homologous recombination. But you can also interfere. So you can use the Cas9 and the guide RNA as a sort of antibody, but that recognizes a sequence of DNA. And you can interfere, decrease the transcription by targeting the promoter, the transcription of the gene, or you can also activate the transcription of a gene. Here is a double nick. Is a strategy to reduce the number of off-target effects, but is less potent compared to this one, for instance. Or you can even purify, because as I mentioned before, the system is sort of an antibody that recognizes the DNA sequence. So what you can do, you can pull down a DNA sequence using the CRISPR and Cas9 system. Is a Cas9 that is mutated, so it doesn't generate a double-strand break, but with the guide RNA binds to the DNA, and then you pull down a sort of chip. But instead of using an antibody, you use the Cas9 system. Or you can visualize, sort of fish. Instead of using a probe made of DNA or RNA, you use the protein with the guide RNA attached. You can screen or you can tag. You don't have the antibody for your protein. You can insert by homologous recombination a tag, MIC, V5, flag, at the end, beginning, wherever you want, the flag in your protein, and then visualize your protein, for instance. So this then now I will go to the second part, so to some examples of our research projects. So I briefly introduce my lab. We are in 5 people, and I'm the PI of the lab together with Milena Nicoloso, and we are involved, as I mentioned earlier, in the discovery and investigation of novel non-coding regions in the epithelial and ovarian cancer predisposition and in the colon cancer aggressiveness. And we use the EMT as a model of colon cancer aggressiveness. And I will show you 2 different strategies. One is a candidate-based approach, so to discover and investigate novel non-coding regions involved in epithelial and ovarian cancer predisposition. We used SNPs, so GWAS studies, to locate regions involved in epithelial and ovarian cancer. And instead, in the second example, I will use an unbiased approach using a transposon-based genetic screen to identify genomic region involved in colon cancer aggressiveness. I will not go through all the aims and the objective of these 2 projects, but just give you some examples on how we use the CRISPR-Cas9 system. Concerning the first, the SNPs involved in epithelial ovarian cancer predisposition, we used this public available database, that is the Genome-wide Association database, and here these are the human chromosomes, and all the dots are different SNPs that have been associated with several traits, with several diseases, okay? And you can also find some involved in ovarian cancer predisposition. And the SNP associated, actually the only SNP associated with ovarian cancer predisposition is located in the 9p22 region of the human genome, and is an intergenic SNP, is located about 40 kb downstream of this gene, that is basonuclein 2, and about 200 kilobases upstream of this other gene that is called centlin. These genes are very poorly characterized, and there is no description of their roles in ovarian cancer. But as you see here, these are all the SNPs that are correlated with this one, and this identifies an haplotypic block. It means that this region of genome tends to move together during evolution. And this haplotypic block includes the BNC2 promoter. So we got interested in the SNP and the BNC2 promoter, and look whether the SNP somehow may affect BNC2 expression. I'm sorry, but the projector slices my presentation. And this is the BNC2 expression in ovarian cancer samples. These are the controls, normal ovary, and these are the tumors. And you can appreciate that the tumor have a lower expression of this gene, whereas for the centlin gene, this gene, there is no difference. And this is the region, a zoom of the region. In blue, it's the location of the SNP, and here the 2 genes, BNC2 and centlin. And we also got interested in this region because exactly in the SNP region, it was described this non-coding transcript, AK024561. And we also look at the expression of this transcript, but is very, very low, okay, almost undetectable. So what we look at were, for instance, histone marks to see whether the SNP was in a regulatory element like a promoter or an enhancer. But there is no signal. So these are 3 different cell lines of ovarian cancer, and this one does not express BNC2, these intermediate levels, and instead COV318 is one of the cells with the highest expression levels of BNC2. And the histone marks for promoters or regulatory elements correlate very well with the BNC2 expression, so high signal in the cell with high protein expression, no signal in of 90 with a no expression. But we could not detect any enrichment in the AK region, so in the SNP region, meaning that the SNP is not contained in a regulatory element, a promoter or enhancer. And so what we decided to do was to generate a knockout by deleting 5 kb regions around the SNP, which also includes the transcript. And so we designed 2 guides RNA, guide 1 and guide 2, targeting this region and this region. And when you use this type of approach, there can be the chance that is not repaired here and here, but the cell get rid of this 5 kb and attaches the 2 ends. And indeed, we found, for instance, some clones where if you do a PCR using the forward and reverse primer here, shown in the picture, in the parental cell, you get this band that is about 5 kb. This is a wild-type clone, and this is a knockout clone when you have about a band of 1.3 kilobases. And when we look in, so we collected a few clones, and as a control, we use clones that were here and here, they had errors, mistakes made by the non-homologous end joining. That was a mark that in that clone, the CRISPR was active, generated a double-strand break, and the mistake here and here was a mark of the Cas9 and guide RNA action. And when we look in the deleted clones, the BNC2 expression in 3 different regions was lower compared to the wild type. So we associated the region where the SNP is involved as a regulator of the BNC2 expression. In the other project involving the colon cancer aggressiveness, we used a transposon-based screening. This is the transposon, is the sleeping beauty transposon with the cassette that you insert and the transposase. We used the ACT116 colon cancer, and the cassette is expressing EGFP. So after you transfect the 2 plasmids in the cells, you get cells that are green. And after keeping the cells in a low adherence culture, most of the cells die, some cells survive. And those that survive are EMT-like, so epithelial mesenchymal transition-like cells. And then if you retrieve the transposon insertion in those cells that are green and that survive to a low adherence, you start to look at whether the transposon inserted in some regions and generated a new phenotype, that is the resistance to the low adherence and acquired EMT-like cells. And we got interested in one of the insertion because it was located in the 3' UTR of a gene that was recently described as a regulator of EMT. And we got interested because it was exactly 1 nucleotide after the seed sequence, the target of a the predicted target of a microRNA. So our hypothesis is that the insertion of the transposon disrupted this regulation. The MIR is an antimetastatic MIR. So by the insertion of the transposon, the protein has bypassed the regulation of the microRNA. And to confirm this hypothesis, we used in 3 different ways the CRISPR-Cas system. The first was to design a guide RNA that was targeting exactly the seed sequence of the microRNA in this way. So you design the guide RNA that is targeting here, then you generate mistakes, errors, by non-homologous recombination and also deletions. Then you screen the clones, and you look for those that have a deletion in the 8 nucleotides of the predicted target site. And since in our department they are now running quite few MySeq experiments, we just use the machine to run our clones. And so this is if you do a target sequencing of this region, and then there is a online software that gives you this analysis for exactly for CRISPR. You get the deletion size. So this is means one nucleotide deleted till 76. So this is the range of deletions that you get when you design a guide RNA for your sequence. So you can see that you have quite big deletions with one guide RNA. In that region, in the size, because the non-homologous enjoining is error prone. So there is no, the deletions are different in size. So it's quite random, the mistake that you generate. Because you were expecting like small or, but actually he reported that you can get deletions. We are now generating more of this data. So these are like the first two that we got. Yes, all. And instead the insertions are mostly one nucleotide, you see? Only one nucleotide. And this is the location of the deletion, and the peak is here, is exactly between the 3rd and the 4th nucleotide where the Cas9 should generate the double strand break. And these are the location of the insertions. That is the same nucleotide. The other approach is to change the target sequence of the microRNA with another sequence, for instance, T7, and do the same experiment with the MySeq. So you get this deletion, this distribution, this insertion, and this peak here. The red sequence is 8 nucleotides. The T7 is 20, 21. And so the difference is about 12. And you get this insertion because the program recognizes it as an insertion, but is actually